Welcome to Circuit Buddy. Today we will be discussing about nucleic acid. In this video lecture, I am going to make this chapter super simple for you. So keep watching the video till the end so that you can understand and memorize all the important topics of this portion and you do not miss any important point. So nucleic acid is a polynucleotide chain. Polynucleotide chain. Okay. Now, poly means many. So, many nucleotides are linked to one another to form a chain, and we call this chain nucleic acid. Okay. Now, you can see that this nucleotide is the building block of nucleic acids. So, we are going to discuss about nucleotide first. So the main components of nucleotides are, number one is the pentose sugar, okay, that is ribose or deoxyribose. Okay, now number two, let me take another color. Nitrogenous base. Okay, and number three is phosphate group. Okay, so let us discuss about the pentose sugar, that is the ribose or deoxyribose first. Okay, so if you draw the structure of the ribose, you will have CH2OH. So you can see that this is the 1 prime, this is the 2 prime, 3 prime, 4 prime and 5 prime. Okay. So this is a 5 carbon sugar. So pent means 5 and ose means sugar. So it's a 5 carbon sugar and this is ribose. Now if you want to draw the structure of deoxyribose, just remove this oxygen from the 2 prime position and it would be deoxyribose. Okay, deoxy means the withdrawal of the oxygen atom from the 2 prime position or any other position. Okay, so it is called the deoxyribose. Now we will be talking about the nitrogenous bases, and for this we have to erase the board. Nitrogenous bases are of 2 types. Okay, they are, one is purine, you can call it A, another is pyrimidine. Okay, now there are 2 purines. Number one is adenine, number 2 is guanine. Okay, pyrimidines are 3 in number. One is thymine, number 2 is cytosine, number 3 is uracil. The first 4 nitrogenous bases are found in DNA, whereas in RNA thymine is not present. Instead of thymine we have uracil. Okay, now we will draw the structures of these nitrogenous bases one by one. So first of all, let us erase the board. So if you draw the structure of a purine, you will have a heterocyclic ring structure. Okay, so you see this is a heterocyclic ring structure. Here's a double bond, another double bond, another double bond, here double bond. These carbons would have another hydrogen. Okay, and there is the NH2 group. Okay, so actually this is the structure of purine. Okay, so first of all let us draw the structure of purine, then we will draw the structures of adenine actually. Okay, so this is the structure of purine, and from it we will be drawing the structures of the nitrogenous bases. You see that it is a heterocyclic ring. Here is a hexagonal ring, here is a pentagonal ring, and these two rings form the purine structure. In case of the pyrimidine, we have okay, so this carbon should have another hydrogen. Here is another hydrogen, here is another hydrogen. Okay, so this is the pyrimidine structure. This is the pyrimidine structure. Now we will be drawing the nitrogenous bases. First we will draw the adenine. So for this, let us draw the structure first. It is the purine structure. Okay, but before that, let me give you a very important information about the numbering of these carbons. Okay, we will be going to that diagram later. So you see that in case of purine, we number the atoms from this nitrogen 1, 2, 3, 4, 5, 6, okay, 7, 8, 9. In case of purine, in case of pyrimidine, we name it as from this nitrogen 1. So this 1, 2. So let me take another color. 1, 2, 3, 4, 5, 6. Okay. So you see that in case of purine, it is going in this direction, or it is called anti-clockwise. Okay, here this is going in this direction, this is called clockwise. Okay, now let us name the adenine. So first of all, let's draw the structure. Here double bond, another double bond. Okay, so you can see that this carbon is the 6th carbon and adenine has an amino group in the 6th position. Okay, and we call this as 6-amino, and this structure is of purine, so 6-amino purine. Okay, so it is also called adenine. Now I will be giving you a very simple trick of drawing the nitrogenous bases. Okay, so first of all, what we will do, we will draw the adenine diagram first, and then the guanine, cytosine, thymine, uracil, these structures. Okay, now if you want to draw the structure of guanine, okay, you will have a double bond O in the 6th position, okay, and the amino group in the 2nd position, okay, and this is the double bond, this is the NH, and it's complete, okay. So this is actually 2 amino 6 oxo purine or guanine, okay. Now if you want to draw the structure of the cytosine, let us draw the structure of cytosine first. Carbon, nitrogen, carbon, nitrogen, carbon, nitrogen, carbon, okay. Now we have this double bond here. Now we know very well that from this nitrogen we will count 1, 2, 3, 4, 5 and 6, okay. So it has a double bond O in the 2nd position, and in the 4th position it has the NH2, okay. And so there is a double bond here, and this is the structure of cytosine, and we can call this structure as four amino 2-oxopyrimidine, okay, or cytosine. Now I will be giving you a very simple trick, but first of all let us draw the structures first, okay. Now we will draw the structure of uracil. So here is carbon, nitrogen, again carbon, nitrogen, again carbon, nitrogen, okay. So the structure of uracil is very simple. In the 2nd and 4th position we have 2 oxygens, okay. So we will have only 1 double bond here. Sorry, there is hydrogens, I have missed the hydrogens, sorry for that. So there is another hydrogen, okay. Now here you see there is a hydrogen, there is another hydrogen, there is another hydrogen, there is another hydrogen, okay. And the structure is 2-oxo, so the structure name would be this structure, okay, 2,4-dioxo or bis-oxopyrimidine, okay, or we can call it the uracil, okay. Now from uracil you can easily draw the structure of thymine, and for that you have to just erase this hydrogen in the 5th position and give a methyl group to it. So give a methyl group to the uracil structure. So carbon, nitrogen, again we have the carbon, nitrogen, carbon, nitrogen, okay. So you have double bond O, hydrogen, again hydrogen, here is hydrogen, and this 5th position we have methyl group. So the name will be 5-methyl uracil, or we can call it thymine. Thymine is found in DNA, not in RNA, okay, and uracil is found in RNA, not in DNA. Now it is a very important thing. Now we will learn to draw the structures in a very simple way. This is the ordinary way, people tell you this simple way, but now I will teach you how you can draw the structures in a very easy way. First of all, let's see, this is the 6-amino purine, okay. I should erase the board, okay. So let us erase the board so that you can better understand. I will draw a bigger structure of adenine, and from that I will be drawing all the structures. So this is the structure of adenine. You know this is a heterocyclic ring structure. Okay, nitrogen, carbon. So this is the structure. This is the hydrogen. Okay, now this is NH2 aminopurine. Okay, now you want to draw the structures of the other nitrogenous bases. For this we will go from adenine to cytosine to guanine to uracil to thymine. Okay, so from adenine we will go to the cytosine. For this, as cytosine is a pyrimidine base, just remove this structure here. Okay, and add just here a double bond O and remove this double bond. Okay, and now you see this is the structure of the cytosine. Now if you want to draw the structure of the guanine, as guanine is a purine base, you have to draw the structure first. Okay, so now you have to exchange the oxygen for the NH2. So here would be, we would have the double bond O, and here we have NH2. So here it would be a double bond. This double bond will vanish. This nitrogen would have another hydrogen. Okay, so this is the hydrogen here. This is the structure of guanine. Okay, now we will draw the structure of uracil. So for that, uracil is a pyrimidine base, so we will remove this structure first. Then we will replace this NH2 with double bond O. So this double bond will vanish, and this is the structure of uracil. Okay, now from uracil we will be drawing the structure of thymine. That's very simple. Just give a methyl group to the 5th position, and it's the thymine. Okay, so these are the various ways you can draw the structures, and please comment if you liked the process, the technique that I used here. Okay, now after that we will be talking about the nucleotide structure as a whole. Okay, the internucleotide structures. So let us draw the structure of the sugar. Okay, so this is the sugar structure. Sorry. Okay, sorry. Okay, it happens. Okay, so you see this is the 1 prime, 2 prime, 3 prime, 4 prime and 5 prime position, and with it the nitrogenous base is linked. Okay, so the nitrogenous base is linked by the 9th nitrogen. Okay, okay, so this is the NH2. So you see this is the structure of the adenine. Okay, so this is the structure of the adenine, and this adenine is linked with this hydroxyl group, removing one molecule of H2O, and this linkage is called the glycosidic linkage. Okay, now in this position we will have the phosphates. Okay, so This OH and H will be removed as H2O, and the bond formation here is the formation of a ester bond, because whenever an alcohol and acid bonds or reacts with each other, ester bonding occurs. Okay, so ester bond or phosphomonoester or phosphoester. Okay, now there may be more than one phosphate, maybe 3 phosphates. Okay, so again H2O removal, again phosphate here, and H OH OH. Okay, again H2O removal. So you see that 3 phosphoester bonds are formed. Okay, and these hydrogens are removed because in the physiological pH it is negatively charged. Okay, so O minus, O minus, O minus. Okay, so this is the structure of ATP or adenosine triphosphate. Now it is a very important nucleotide. Okay, so in a nucleotide we have the nitrogenous base, the sugar and the phosphate. Okay, now the nitrogenous base plus the sugar is called the nucleoside. So it is very simple to remember: N nitrogenous base and S sugar, NS nucleoside. Okay, and when you add the phosphate to it, it would be nucleoside, monophosphate, diphosphate or triphosphate. So how many nucleoside triphosphates are there? So there are many types of nucleoside triphosphates, that is the ATP, the adenosine triphosphate, the guanosine triphosphate, the cytidine triphosphate, the thymidine triphosphate, and the uridine triphosphate. Okay, there may be diphosphates also: the adenosine diphosphate, guanosine diphosphate, cytidine diphosphate, and thymidine diphosphate, and uridine diphosphate. There may be monophosphates also, monophosphates also: adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, thymidine monophosphate, and uridine monophosphate. Okay, so these are the various types of nucleotides present in the cell. Now in the next video we will be talking about DNA in details, and in the third video we will be talking about RNA in details. Thank you very much for cooperating with me. Thank you very much once again for watching my videos. Please like, comment and share if you like the video, and goodbye.